Good morning, good afternoon, good evening. This is Dr. Jenkins. We are here for our third lab. And the first part of this lab is going to be the microscope lab. So I just want to go through some of the parts of this lab so you are clear on what it is I'm asking you to do. Sound good? Sounds good to me. Let's go with red today. Yes. Okay. The first part of the lab is just to learn the parts of a compound microscope. I've given you a list of things that I'd like you to know. I'm not going to sit here and go through all the parts. There will be some pictures that you can review on Blackboard, but I think you will learn the best by just sitting down yourself and memorizing these parts. There are, oh, actually, you know what? I made a little mistake. Okay. These are the parts of the microscope right here. This box is terms that we're going to talk about. But the parts of the microscope are here. Make sure that you review them. And yeah, if you have any questions, let me know. Now let's talk about terms. We can see here some terms, and let me just go through some of them for you. Resolution is really the same thing as clarity. A microscope is designed to magnify things so we can see it better. And the better the microscope, the more resolution it has, the more clearly it will show the objects. You can think about a camera, the better the camera, the more clear the images are going to look. Don't worry about resolving power. Let's talk about real versus virtual image. The virtual image is what you end up seeing when you look through the ocular lenses. And by the way, the ocular lenses are right here. What you see looking through the ocular lenses. It's called a virtual image because when the image goes through the ocular lens, it then is magnified by the objective lenses, okay? And the objective lenses are the set of lenses on a little rotating thingy here. So what you see is actually a virtual image because when the ocular and the objective lenses work together, it actually alters the image a little bit. And when we're in the lab, you can see this a little better because the actual slide that you have here, when you look at it through the ocular lens, what you're seeing is upside down and reversed. And that just has to do with the interplay between the ocular and objective lenses. So the virtual image is the adapted image that you view. And it's considered virtual because it has been manipulated by how the ocular and objective lens work. The real image would be what the real image is. So if you were just to look at the microscope slide with your eyeball. Okay, let's look down here. The compound microscope is this picture of the microscope. And it's the kind of microscope we use in lab. And that'll be the kind of microscope that I will have pictures on Blackboard for you. We are going to define working distance in a little bit. We're going to define field size in a little bit. And oil immersion is something that can be used with the 100 power objective lens. So sometimes if you're viewing a live specimen, if you take microbiology, you'll definitely be using some oil immersion. Sometimes along with that high power objective lens, you put a drop of oil into the specimen. And it just makes it easier to see live specimens at a high power. We are not going to be using oil immersion in this class, but I just want you to know what it is. All right, so after you learn the parts of the microscope, and after you review the images that we've talked about, then you're going to go ahead and do the lab. And there's a couple exercises. Obviously, these will be better to do if we were in person, but we're not. So I've tried to make it as good as possible. So you're going to review the magnification. And here's the information. The ocular lens itself magnifies the image 10 times. And then with the objective lenses, there are 4 different objective lenses. Usually it's a 4 power, 10, 40, and 100. So to determine the overall magnification, you're going to multiply the ocular, which is always 10. You're going to multiply the ocular times whatever the objective lens is. Pretty simple, okay? And that'll get you the total magnification. I'm going to have you fill out the summary table, and the first thing that it's asking you is to write in the total magnification. So now you'll be able to do that. Then we're going to talk about working distance. Make sure you review the definition. It's pretty simple, my friends. Working distance is the distance between the bottom of the objective lens and the top of the microscope slide. Usually we do this in person, but I've set it up so you can measure the working distance using these handy dandy rulers for all of the different objective lenses. And you can see what happens to the working distance when the power of magnification goes up. And of course, we're going to be recording all of our data this week in millimeters. So on this ruler, each individual line is a millimeter. And when you get to here, that's going to be 10 millimeters or 1 centimeter, but we're going to use millimeters. Not so bad, folks. Now we're going to talk about field size. When you look through the oculars, you're going to see a field of vision. So whatever it is you're looking at, it's going to be viewed in that circular field of vision. Well, sometimes it's helpful for us to measure the field size, which is the width. Because let's say you have a specimen, and that specimen is that size. If I know the field size, I'm going to make up a number just because I want you to do the actual measuring. If this actually was 100 millimeters, it's not, but if it was, and this object I have is about half of the width, then I could estimate that the specimen I'm looking at is 50 millimeters. So measuring the field size or the field diameter can help us estimate the size of objects if we wish. So I've got a field size here, and I'm going to ask you to measure the diameter at all of the different objective power lenses, and both the working distance and the field size, also in millimeters. You're going to enter that data into this table, working distance and field size. The last thing that you will look at involves depth perception. So you can read through this, but let me give you the highlights. Some of the specimens or the slides that we use are thick. Some of the, like for example, our next 2 labs are going to be on histology. Well, if we take a sample of the skin, for example, it's more than 1 layer of tissue. So we have to understand that some of the specimens that we have will be multi-layered. And because they're multi-layered, it's helpful to be able to determine the different depth perceptions. So this is going to be an exercise that we usually do in lab, but I'm going to have you do just with the pictures. And it involves crossed threads. So imagine we have one microscope slide and it's got 3 different color threads lying on top of each other. So even though at first you can't identify the difference, in reality, one of these color threads is on top, another color thread is on the middle, and another color thread is on the bottom. So here's how we're going to understand depth perception. And in the end, we're going to be able to identify which color thread is on the top, which is in the middle, and which is on the bottom. So we're going to start on this left one, okay? And we can see here the image that is in focus first is actually on the bottom. Huh? I'm recording. All right, so I want you to first look at the picture on the left. Sorry about that. If you look at the picture on the left, I want you to identify, and it can be a little bit difficult, but feel free to zoom in. I want you to identify which color thread is in focus. So we've got yellow, red, and blue. And if you look closely, you should be able to see that one of those color threads is more in focus than the other. So whatever color that is, that's going to be on the bottom. Now we're going to move over to this one. And what these different views are looking at is, after we found the first one on the left, then I started to move the stage further up. And then this second one came into focus. So you're going to see, out of the three colors, which one do you think is most in focus? And then of course we're going to see the last one, as I move the stage even further up, which one came into the most focus. Okay? And then you're going to write your answers based on that, which color thread is on the top, which is on the middle, and which is on the bottom. Excellent. Then you have some review questions. This in total is worth, what did I say, 24 points? All right? So good luck, everyone, and if you have any questions, let me know.